My name is Maral, and today I'll be talking about my technical contributions to our Capstone project, which is in producing viral vectors for gene therapy. Gene therapy is a way to use genetic information to treat or prevent disease. Vectors are the vehicle that delivers genetic material into target cells, and they essentially act like an envelope that carries genetic information. With gene therapy, we have the potential to save $45.6 billion a year in Canada. And here at GetGenix, we will be producing lentiviral vectors, or LVs for short. To comply with safety and regulatory requirements, we must separate out contaminants such as DNA and proteins. Anion Exchange Chromatography, the unit I'm responsible for, is the main protein purification step in our process. As LVs, proteins, and DNA are fed into the column, AEX uses a positively charged resin to attract the negatively charged LVs while eluting or flushing out the weakly bound proteins and DNA. However, to implement AEX column into our production process, we must ensure that the column we need is available to buy, operates within a reasonable amount of time, and is economically feasible. And these three factors all depend on what column size do we need to process the feed. So to size the AEX column, I used the Klinkenberg equation, which models non-ideal fixed bed adsorption. Along with the assumptions listed, this equation uses linear driving force model, so a linear adsorption isotherm was needed to model LVs. To prevent shear on vectors, the AEX column is often operated at low flow rate, which results in low Reynolds number. So to calculate external mass transfer resistance in the column, I found a correlation by Reichelt et al. That was valid for my system. Additionally, pressure drop along the column was modeled by the Ergen equation. And MATLAB was used to model the column. Along with the equation, the major inputs affecting my model were the linear isotherm, choice of resin, bed porosity, and flow rate. So, using the Klinkenberg equation, the code generates breakthrough curves that I can use to calculate operation time and amount of LVs absorbed. Due to the limited data on LVs, I used the linear isotherm for water viruses. As can be seen from the graph, I looked at 4 available commercial bed volumes and their breakthrough curves. From the curves, the commercial bed volume that can process our feed is 260 mils, and it will take roughly 40 minutes to run the feed and elute the vectors. Also, it's important to note that pressure drop was a limiting factor for my simulation because the design pressure of the commercially available column is 4 bars, so I had to limit pressure drop to roughly 2 bars. So from the simulation, we were able to determine that the bed is commercially available, operates within a reasonable amount of time, and is economically feasible. Thanks, and for further questions you can reach out to me or take a look at my references.